Welcome back to the Cell and Molecular Biology course. I'm your course instructor Rimya Pendral. In this video, we will explore the Golgi apparatus, a crucial organelle responsible for modifying, sorting, and packaging molecules inside the cell. Often referred to as the post office of the cell, the Golgi apparatus ensures that biomolecules are sent to their correct destinations and thus supporting essential functions in both animal and plant cells. By the end of this video, you will have a comprehensive understanding of its structure, functions, and importance in cellular processes. Now let's begin. The Golgi apparatus, also known as the Golgi complex or Golgi body, is a vital membrane-bound organelle in eukaryotic cells. In the illustration, you can see an arrow mark pointing out the Golgi complex. This organelle plays a fundamental role in the processing and transport of proteins as well as lipids, uh, and it ensures that these biomolecules reach their appropriate locations within or outside the cell. This complex was discovered by Camillo Golgi in 1898. This organelle is essential for maintaining cellular organization and efficiency. Without the Golgi apparatus, cells would struggle to manage their internal functioning, leading to a breakdown in essential cellular functions. So next we will see the structure of the Golgi apparatus. The Golgi apparatus consists of flattened membrane-bound sacs called cisternae, which are stacked together like a pile of pancakes. It has two distinct phases: the cis phase, which receives newly synthesized proteins and lipids from the endoplasmic reticulum, and then the trans phase, which ships these processed biomolecules to their final destinations. These compartments work in a organized manner to ensure that proteins and lipids undergo necessary modifications before they are dispatched. So in the illustration, you can see the cis phase and trans phase, and then you can see the cisternae, which is, as we have learned, it is a stacked structure, which is like a pile of pancake. And then you can see the incoming transport vesicle and newly formed vesicle, then secretory vesicle. As we have seen, the cis phase receives the incoming transport vesicle, which are the lipids or proteins from the endoplasmic reticulum, and then it get processed and from the trans phase, it is released as a secretory vesicle. Now let's see the functions of Golgi apparatus. One of the primary functions of the Golgi apparatus is to modify the proteins to ensure they function properly. Now let's see it in detail. After proteins are synthesized in the rough endoplasmic reticulum, they enter the Golgi where they undergo glycosylation, the addition of carbohydrate chains, or phosphorylation, where phosphate groups are added. These modifications affect protein stability, target, and function. For example, Glycoproteins play a key role in immune recognition, while phosphorylated proteins are crucial for cell signaling pathways. So the precise modification of proteins by the Golgi is essential for the health and proper functioning of an organism. Besides proteins, the Golgi apparatus is also responsible for lipid modification and transport. Here is an illustration showing the lipid processing and transport involving the Golgi and endoplasmic reticulum. Lipids are essential for cell membrane integrity and hormone synthesis and cellular communication. The Golgi modifies lipids to create molecules such as sphingolipids, which are crucial for cell membrane stability and function. Additionally, it sorts lipids into specialized vesicles that transport them to various parts of the cell. Without this system, membrane formation and maintenance would be compromised, leading to severe cellular dysfunctions. Once proteins and lipids have been modified, the Golgi apparatus packages them into vesicles, which serve as transport carriers within the cell. There are different types of vesicles, including transport vesicles that move materials within the cell, and the other type is secretory vesicles that release the molecules outside the cell, and lysosomal vesicles are the ones that contain digestive enzymes. The accuracy of this packaging and transport system ensures that these biomolecules reach their destinations efficiently and to maintain cellular order and functioning. Now let's see the lysosome formation. The Golgi apparatus also plays a crucial role in forming lysosomes, which are cellular organelles responsible for breaking down waste and recycling cellular components. These lysosomes contain hydrolytic enzymes that help degrade old organelles, foreign substances, and also cellular debris. This function is essential for maintaining cellular health and prevent the accumulation of toxic waste within the cell. Without the proper functioning of lysosomes, cells would struggle to remove damaged components, which leads to conditions such as lysosomal storage disorders. The Golgi apparatus is vital for the secretion of proteins and lipids outside the cell through exocytosis. For example, hormone secreting cells rely on the Golgi to package the hormones like insulin for release into the bloodstream. Similarly, neurons depend on the Golgi to package the neurotransmitters, ensuring proper communication between nerve cells. The Golgi's role in exocytosis highlights its importance in intercellular signaling and overall the functioning of an organism. In plant cells, the Golgi apparatus has an additional function, synthesizing polysaccharides like cellulose, which are critical for the building of the cell wall. The rigid structure of the cell wall, which provides strength, support, and protection to the plant cells, uh, which also enables them to grow and maintain structural integrity. Without the Golgi's contribution to cell formation, that is cell wall formation, plants would lack the ability to maintain their shape, and they would not be able to withstand the environmental stresses.
So the Golgi apparatus is essential for protein and lipid modification,
As well as vesicle transport, lysosome formation, and also secretion of uh, proteins and lipids. That is, example, the secretion of neurotransmitters like acetylcholine, and then for secretion of hormones like insulin. For all these functions, the Golgi apparatus is essential in eukaryotic cells. It acts as a central hub for cellular logistics, thus ensuring that biomolecules are correctly processed and delivered. Without the Golgi, the cell's ability to function efficiently would be severely impaired. So, in the illustration, you can see the transport vesicles bringing molecules from rough endoplasmic reticulum, and then the molecules fuse with the membrane and are sorted based on destination. And then these molecules undergo remodeling and modification in cisternae. And finally, the modified biomolecules are secreted either out of the cell or within the cell. Golgi dysfunction has been linked to various diseases, including neurodegenerative disorders like the Alzheimer's, Parkinson's, as well as certain genetic conditions that affect protein processing. All these are linked to Golgi apparatus dysfunction. So, research into Golgi-related diseases is helping scientists to develop new treatments to address these cellular malfunctions. In summary, the Golgi apparatus is a critical component of the cell's system, ensuring proper protein modification, lipid transport, lysosome formation, and secretion. Its role in cellular health is indispensable, and undergoing research continues to reveal its importance in disease mechanisms. So, so understanding the Golgi complex gives us valuable insights into the intricate world of cell biology. Thank you for watching. See you in the next video.